Good morning, everyone. Today I'm going to tell you a little bit about Kinoxis Therapeutics and our drug discovery and development programs. So Kinoxis is a private Australian-based biotech company developing small molecule inhibitors of ALOX15. So ALOX15 is an intracellular enzyme that's of increasing interest in a number of different CNS conditions, with our particular focus areas being the treatment of agitation and aggression and central modulation of pain. Our lead compound, KNX100, is currently in an active phase 2 clinical trial for the treatment of agitation associated with dementia. And in addition to KNX100, we have a next generation of highly optimized ALOX15 inhibitors with the lead from that next generation series ready to enter IND-enabling studies. We're currently looking for the right partner to help us take our assets through development. So I want to take a moment now just to tell you a little bit about how we came to be focused on this target, ALOX15. So our phase 2 compound, KNX100, was actually discovered through a phenotypic screen. And in parallel to developing KNX100, we conducted a comprehensive multi-year target deconvolution program, which identified the primary target for KNX100 as ALOX15. And this then enabled us to generate this suite of highly optimized small molecule ALOX15 inhibitors that we refer to as our next generation compounds. So what is ALOX15? It's an intracellular enzyme. It's expressed in brain regions that are of considerable interest to us in both glial cells and neurons. And it oxidizes polyunsaturated fatty acids into these bioactive eicosanoids. Now, these eicosanoids act as fast signaling molecules, regulating neuronal excitability and synaptic strength, and they are also lipid redox mediators and play a key role in neuroinflammation and neuroferroptosis, which are core drivers in multiple CNS pathologies. And if you look at the rising publication volume on ALOX15 over the last 10 years, this really underscores the increasing interest in ALOX15 biology, as well as in its potential as a therapeutic target, including in areas that are of considerable interest to Kinoxis Therapeutics. And one of these is the treatment of agitation and aggression associated with dementia. So this agitation and aggression are among the most challenging and prevalent symptoms of dementia. And unfortunately, the current treatment options are extremely limited. There's only one FDA-approved treatment for agitation associated with dementia. That's the atypical antipsychotic brexpiprazole, and it has some really serious safety concerns, including a box warning due to increased risk of death in the elderly with dementia, the very population that it's being used to treat. And the clinical development pipeline is really dominated by repurposing and reformulation. There's not a lot of novel mechanisms. And when you look at the clinical pipeline, these drugs share at least some of the same safety concerns seen with the approved atypical antipsychotics. A key point of differentiation of our Kinoxis ALOX15 inhibitors is that they avoid these major safety liabilities of the current treatments: no sedation and no cardiovascular risks. Another key point of differentiation is obviously our mechanism of action. There are no approved ALOX15 inhibitors. We know that ALOX15 is upregulated in the brain in Alzheimer's and dementia, as well as the products of ALOX15 metabolism. It's also upregulated in mild cognitive impairment, this early prodromal phase where you start to see the emergence of agitation and aggression. And ALOX15, from a biological perspective, really sits at the crossroads of multiple pathogenic processes in Alzheimer's and dementia, making it a very compelling target. When we look at the neural network and neurocircuitry level, inhibiting ALOX15 has the potential to provide much more effective regulation of the core neurocircuitry underlying aggression than the current atypical antipsychotic treatments. So here we used a technique called CFOS-I-DISCO to conduct unbiased whole brain activity mapping. We identified a neural network associated with hyperaggressive behavior in mice. This mapped on really closely to the very well conserved neurocircuitry that underlies aggression across species. And what was really striking is when we treated these mice, these hyperaggressive mice, with our ALOX15 inhibitor KNX100, it normalized or partially normalized activity across 44% of this aggression-associated network. And it was specifically targeting the circuits at the higher order level that underlie the psychological mechanisms that drive agitation and reactive aggression. In stark contrast, the atypical antipsychotic risperidone, which is approved in many countries for the treatment of agitation associated with dementia, really failed to meaningfully engage the aggression-associated network, and it primarily had effects on circuits outside of the aggression network that are involved in motor control and arousal. And this is highly consistent with what we see at the behavioural level, where our Kinoxis ALOX15 inhibitors are able to inhibit hyperaggressive behaviour without any sedation at all. In contrast, atypical antipsychotics are only effective at sedating doses, and actually at low doses we see paradoxical disinhibition and an increase in aggressive behaviour, which is also seen in a subset of the clinical population. We've shown a strong class effect with multiple highly structurally distinct ALOX15 inhibitors able to inhibit hyperaggressive behaviour without sedation. And we see really consistent effects across different models. So the chronic isolation-induced aggression model in mice, the APP-PS1 genetic mouse model of Alzheimer's disease, and a rat model of drug withdrawal-induced aggression. Whether or not this translates, we'll find out, and we've got our first shot at that now in our active phase 2 clinical trial in agitation associated with dementia. It's a randomized placebo-controlled trial. Our primary outcome measure is the Cohen-Mansfield Agitation Inventory, and we're also looking at a number of other measures of agitation and its impacts. The trial is recruiting at the moment, and we expect to complete recruitment by the end of the year. So turning now to our other therapeutic area of interest, pain, and in particular chronic pain, which affects 1 in 5 people globally. It's already a massive market and it's growing quite rapidly. There are several issues with the current treatments for pain, but an overarching issue is that at best, they provide only very minimal efficacy for the management of chronic pain. And ALOX15 is a really compelling target for the treatment of pain, including chronic pain. And inhibition of ALOX15 provides the opportunity to inhibit 2 critical pathways in pain: a glial-driven NSAID-resistant pain pathway and a reactive oxygen species-driven pathway that's increasingly being implicated in the chronification of pain. There's lots of examples in the published literature of ALOX15 inhibition reducing pain across a variety of different models, but I want to share with you just one in-house data set, which is from the gold standard chronic constriction injury model of chronic neuropathic pain. So in this model, rats show a chronic pain phenotype indicated by heat allodynia and cold allodynia that increases over the 4 weeks post-injury, stabilizing from 4 weeks. When we treat these rats with an acute dose of KNX100, it very nicely reduces heat and cold allodynia, but perhaps most strikingly, when we provide chronic treatment with our ALOX15 inhibitor KNX100, it reduces pain levels by 70 to 80%, which is sustained across this multi-week treatment period. So wrapping up now, Kinoxis is developing inhibitors of ALOX15, which is a highly biologically relevant target in both dementia and pain. Our comprehensive preclinical data set and improved safety and tolerability profile relative to current treatments, we think holds a lot of promise. Our lead asset, KNX100, is currently in phase 2, and our lead from our next generation series of highly optimized ALOX15 inhibitors is ready to enter IND-enabling studies. Thanks for listening, and if you're interested in learning more, please don't hesitate to come and see me or reach out.